So the first one is given by Agnieszka Burzynska. And Dr. Burzynska is an associate professor at Colorado State University. And she will be talking about metabolic syndrome and adiposity relate to lower myelin and axonal content in healthy adults. The floor is yours.
Hi, everyone. Thank you so much for having me here. It has been really a treat so far. Very exciting conference. And yeah, I'm not a trainee by definition, but I still feel a trainee in the field of QMR because today it's my pleasure to present the first data set we acquired with those more advanced, newer techniques. So first of all, I wanted to highlight this is a collaborative effort, and we are supported by the Alzheimer's Association and NIA. And in this 10 minutes, I'll not be able to cover all details. So my talk is based on our 3 recent publications, some published, some still in the works. So if you have any questions, please email me and I'm happy to provide any details. So the motivation behind our work is to understand the neurobiological processes that are behind cognitive decline in both normal aging and dementia. And our ultimate goal is to understand them to the extent we could postpone, prevent, and maybe even at some point treat cognitive decline. And this year brought some exciting news, especially for the patients and their families. As you may know, there is a drug that was approved by FDA that is addressing the hallmark of Alzheimer's disease, namely amyloid plaques. However, despite the excitement, we know that the clinical trials show that it has only limited effects on the clinical outcomes, and it may only be effective in the early stages of the disease. So we are definitely not yet done with our quest to combat cognitive decline. And that's why in my laboratory, we focus on the alternative pathology related to cognitive decline in both aging and dementia, which is the structural and functional deterioration of myelin. And in the past, maybe 3 decades since there was active research using MRI in that field, I would say that about 95% of studies still use the DTI model. And the newer methods are a bit slowly infiltrating the field. So I'll be presenting results from our newest study, with which we revisit some of the more basic and more complicated questions that have been asked before with DTI, but because of the limitations of the DTI model, they have been a bit inconclusive. So the questions I'll be addressing today are the age and sex differences in myelin and axonal content, what are the preventable risk factors for their health, and whether there's a link to cognition. Because we kind of assume that deterioration of axons and myelin would lead to cognitive decline, and this is the basis of the so-called structure disconnection hypothesis in aging and dementia. However, the in vivo evidence is still very scarce, as you could hear yesterday, for example, in a talk by Dr. Gong, that only using those more specific methods in living humans, we are only kind of finally able to ask those questions. So in our study, we collected data from almost 150 people from the age of 20 to 80. And after the initial recruitment that was pretty strict, we had some additional exclusion-inclusion criteria to reduce possible MCI or early vascular dementia. We invited our participants for visit 1, during which we again measured their blood pressure, and this was one of the exclusion criteria. And this criteria could see that the flare scans looked really much better. So this was kind of main reason to exclude our participants. And after this, they underwent a very comprehensive cognitive testing. And after 1 week of wear of physical activity monitor, we invited them for about 1 hour scan that was composed of several different MRI methods. So we applied multi-shell diffusion imaging, and we used the NODDI model so far. But after hearing the talks, I think we need to expand to different models. We also had myelin water imaging, quantitative magnetization transfer, and some susceptibility imaging. But as this sample is still undergoing analysis, today I'll be only talking about the intracellular volume fraction derived from NODDI as a measure of axonal content, and myelin water fraction as a proxy for myelin content. And because our sample included this broad age range, which is related to anatomical differences, we used TBSS to focus on our analysis on the centers of deep white matter tracts. And finally, we extracted regional volumes from those skeletonized representations of the white matter. And our first finding was the expected negative association between age and both myelin and axonal content. In most of the regions, as you can see here, the whole white matter and the prefrontal white matter, we observed the inverted U-shaped function with the peak of both axonal and myelin content occurring at about age of 40. And in other regions, such as the geno of the corpus callosum, which is known to be in particularly affected by aging, we see more linear decline starting already at the age of 20. And we found sex differences only in the ICVF, where men had greater intracellular volume fraction than women, despite of age. We also found significant positive associations between myelin and axons, which are not surprising, and they varied by the regions. So to summarize this first part, something I did not show explicitly, we found that the axonal and myelin content was greatest in the early myelinating regions, so consistent with the developmental theories, but the late myelinated regions showed the most pronounced different changes with age. This is consistent with the emerging literature using different QMR techniques and different samples. The slightly kind of newer finding of this close relationships of myelin and axons is also consistent with the physiology and the trophic support between axons and myelin. So our next question was, are there any preventable risk factors that relate to white matter health? And we focused on the metabolic syndrome, which is a very, very common condition among people. It affects about a quarter of global population and is defined as the co-occurrence of at least 3 of those conditions listed here. It not only predicts mortality, higher risk of stroke, but it is also one of the main risk factors for Alzheimer's disease. And although it has been associated with reduced brain volumes, for example, the underlying microstructure changes are not yet understood. So we collected measures of metabolic syndrome components from our participants, and despite our strict criteria on the blood pressure, also BMI and other health factors, we still managed to replicate that about 22% of our sample had possible metabolic syndrome. Only 50% was lean by the definition of the BMI, and waist circumference is much less forgiving, and only 12% actually were classified as lean, which was even a surprise to us, but it also shows how prevalent metabolic syndrome risk is in our population. And for further analysis, we used metabolic risk score as a kind of continuous score of this risk factor, and also a subscore for adiposity that was combined waist circumference and BMI. So to cut the long story short, we did find the expected negative association. So the greater metabolic risk score or adiposity score, the lower the myelin was. And this effect we observed especially in the more vulnerable, more late myelinating regions, controlling for various demographic and socioeconomic variables that are known to be related to metabolic risk score diagnosis. And our next question was, could we replicate this association with the estimate of axonal volume? And the short answer is no, we did not find similar associations between axons and the metabolic risk score. So we also asked whether those relationships that you can see here, would they still be significant after controlling for intracellular volume fraction, knowing that both myelin and axons were related to each other and related to age. And the short answer is that this relationship was seen to be independent of axonal volume fraction. So to summarize this part, we found that both metabolic syndrome and adiposity were related to lower myelin in those almost above average healthy adults. And whether this is specific to, whether it's really specific to myelin and whether the axons are not involved, we can't yet conclude, but we speculate that axons could be involved at later stages of metabolic syndrome, for example, when people have had it for a longer time. Our finding that the late myelinated regions are most affected is also consistent with, for example, the specific vulnerability, such as lower oligodendrocyte-to-axon ratio. And definitely those findings need to be replicated using maybe better estimates of axonal volume. And now the last part, we also investigated the link between myelin content and cognitive function. And we found that the prefrontal and the geniocorpus callosum myelin was a significant predictor of memory after controlling for age, sex, and education. We also found some associations with processing speed, but they are not that prominent, so I'm just showing you here for the memory. We also use structural equation modeling to show that the myelin in the prefrontal and the geniocorpus callosum mediated the effect of age on memory, which was strong and negative as we expected. So to summarize, our data, I think, shows that we can get very valuable in vivo support for different hypotheses of cognitive decline, such as the disconnection hypothesis. And also that visceral adiposity could be a possible target for new interventions. And here I made this very oversimplified plot of possible myelin kind of trajectory across the lifespan, where the black curve represents someone with some metabolic diseases or challenges, and the green one is characteristic of the a more healthier individual. So it is possible that because our relationships were independent of age, that people with some metabolic challenges may not really reach the peak in their 40s as they could. So maybe the interventions in a early adulthood, not as we typically do in the late or middle adulthood, could be really more successful in helping people reach that peak of myelin to a greater extent that would be protective against subsequent cognitive decline, because we also show that link between myelin and memory. So what will be the next steps are definitely longitudinal studies using QMR techniques that should be also multimodal, because as previous speakers mentioned, there is quite some kind of cross-contamination between and limitation to specificity. So applying different methods in conjunction could give us more information. So not only multimodal, but also multimodal, especially when it comes to diffusion. We are also interested in looking into iron and also to superficial white matter. So thank you very much for your attention. And I also would like to thank my co-authors and other collaborators, especially John Colley and Phil Lee, for helping us learn the new methods. Thank you.
Okay, that was great. Thank you so much. Olivier, do we have questions for Dr. Burzynska?
Not yet. There's no questions in that chat. Maybe I'll ask a question then. So, did you use, I'm sorry if I missed it, but did you use a particular parcellation for looking at the myelin water fraction in the white matter by track, for example, or something like that?
Not sure if I understood your question. Yeah, we did like regional analysis. We used, I was not able to present all regions, but we analyzed about different 16 subregions of the deep white matter. I don't know if
so by white matter tract?
Yeah, we didn't really use tractography. We used this kind of more skeletonized white matter representation. Yeah, so we didn't really use by tract, more by white matter region, just because sometimes I find it really, the result of tractography may vary greatly between ages. So we like to kind of I found that the most replicable, robust results come from these regions where we can identify them in an anatomical way more easily.
Yeah, for sure. Okay, maybe one more question. So, Sudhi Mohalemian asks, how did you calculate the mediation of myelin and age association?
Yeah, so this is actually work done by my postdoc Andrea, who's also here in the audience. So we use structural equation modeling for this part of analysis. So I don't know if we can provide you more details on that. Maybe by email, but I think it was a standard model.
Okay, thank you.